Hello there, glad you can join me today. Right, today I'll be going through the thingy of transcriptional control, the controls of transcription. Now this is specification OCR, and it will be 6.1.1b that I'll be going through today. So, here's what you should know. In every cell that contains a nucleus, the entire genome is present, even genes not required. When the genes are needed, maybe due to a change in the environment, they are expressed and turned on. These occur due to control on three levels. You'll first have transcriptional control. Now, transcriptional control occurs in itself in three different ways. You have chromatin remodeling. This is when DNA is wrapped around a protein called the histone, so it is packed into the nucleus, forming a complex called chromatin. Transcription takes place during interphase, not cell division. This allows euchromatin, loosely wound DNA, so you've got chromatin and you've got euchromatin, which is loosely wound DNA, to be transcribed. As it occurs during interphase, proteins are given the necessary time to be made. Alright, so that's the first bit. Then the second part of transcriptional control is histine modification. So due to histone being positively charged and DNA being negatively charged, the DNA coils tightly around the histone. But if histone is made more negative, the DNA will loosen and allow some genes to be transcribed. So if genes could not previously be transcribed, if it was add a way to make the histone more positive, or more negative in this case, sorry, then more repulsion would occur, and it wouldn't coil as tight, and then giving there space for proteins to transcribe it. Alright, and now the third one. This one is only found in bacteria, such as E. coli. This is the lac operon. Glucose is the main respiratory substrate, but if it isn't present, the E. coli can switch to lactose if lactose is present, and therefore utilize lactose. Now, the bacteria can be very efficient as they only synthesize needed enzymes and place functionally related genes next to each other. There are two structural genes necessary to break down lactose. You have lactose permease, which will code for a transport protein, and it will absorb the lactose. And you've got beta galactosidase. Now that actually hydrolyzes the lactose. Alright then. Now, I have drawn out a little bit of a diagram. So this bit here is your lac operon. Now this R, that is the regulatory gene. Now the regulatory gene is not part of the lac operon, but it does code for the repressor protein. This here, that's the promoter region. This is where RNA polymerase will bind. This here is the operator. And then you've got that one here is the beta galactosidase, and this is your lactose permease. These are the two genes which will code for those two particular proteins. Now, normally, I don't know if I've right. Yep. Alright, so how does the lac operon work? Alright then. Well, here's how the lac operon works. You will have, if glucose is absent, but lactose is present, bacteria can use lactose as a respiratory substrate and break it down to glucose and galactosidase. The repressor protein, coded for by the regulatory gene, as I mentioned earlier, which isn't part of the lac operon, binds to the operator region and covers the promoter region, so the RNA polymerase cannot bind, preventing mRNA being produced, and hence the enzymes involved in using lactose being produced. However, lactose binds to the repressor protein, changing its shape so it is no longer complementary to the operator region, preventing binding and uncovering the promoter region. This allows RNA polymerase to bind and code mRNA, creating the enzymes. So when you've got your repressor protein, it'll be a certain shape, and it will bind to, if I correct, it will bind to the operator, and when it binds to the operator, it will cover the binding sites for the promoter. Therefore, it can't make any of the enzymes required. However, what can happen is that lactose will be a complementary shape to the repressor protein, and once it binds, it will change the shape of said repressor protein, meaning it can no longer bind to your operator, uncovering the promoter region, allowing RNA polymerase to bind. Alright, so the other two levels. You've got post-transcriptional control, which is within a gene. So within a gene, there are non-coding regions of DNA called introns, and coding regions of DNA called exons. All DNA of the gene is transcribed. This is primary mRNA. So primary mRNA will include both the introns and the exons. The introns are removed and exons are joined together to form mature mRNA. Now, not only that, some mRNA can then also be edited through base addition, deletion, or substitution. So if it's not exactly right, it can be altered. And now the last transcriptional level control that you need to remember is post-translational control. So the last one, so we've had transcription, post-transcription, and now we've got post-translation. Post-translation level. Synthesized proteins need to be activated to work. This is done by adding a phosphate group. The process is started by hormones binding to receptors. This activates a G protein, which then will activate adenyl cyclase. Adenyl cyclase catalyzes reactions which turn ATP into cyclic AMP. Cyclic AMP then alters the 3D structure of protein kinase A, activating it. Activated protein kinase A then catalyzes phosphorylation. And that is all of the transcription controls, all kind of explained.